What's up, hippie bio penguins? So as requested, we are doing free response Fridays. So I'm going to try to do them at 2 o'clock, but don't worry because this will always be recorded and posted on my Instagram, like IGTV. It will also be downloaded and posted onto my YouTube channel, and it will also be posted onto the Weebly. So you have 3 different places that you can access this video afterward if you are not able to attend the lives when I do them. Hi, welcome everyone. What's up, baby bio penguins? So we are going to do 4 different questions today. So the 4 questions we're going to do is going to be the 2019 exam, number 8, the 2018 exam, number 6, the 2017 exam, number 8, and the 2013 exam, number 6. Okay. And so the questions were picked based on units. So if you kind of think about the way we've been doing our units, we're currently on Unit 2. So I picked questions that were in Unit 2. And I believe by next week when we have our next FRQ Friday, we'll be in Unit 3. So I tried to kind of put a lot of questions today just so that we would make sure we got a bunch of the Unit 2s. Okay. My advice is that with these FRQ Fridays that you do the questions ahead of time so you kind of have an idea of what the question is and you know what answers you would put if it was you doing the FRQs, because it doesn't really help you to be told the answers. It's best if you are checking your answers and seeing, okay, did I have the logic correct? And all of these free responses are coming straight out of the review guide. So as always, apbiopenguins.weebly.com. There's a huge review guide posted for you. So just go on there and you can download it. And you can actually, it's clearly labeled which years are which. So without further ado, let's go ahead and get started. And if you have any questions along the way, just you can pop them down in the little question box. And I should be able to navigate both. I'm just going to go through those 4 free responses and discuss them as we go. So this free response is number 2019, number 8. You see that we have these 2 different things. Now remember, you want to always think about the diagrams, right? So what does the diagram show me? Okay, so if you look here at this diagram, I've got a bud and I've got an open flower. I can see that in the bud, my vacuole is smaller, and over here I can see I have a larger vacuole. And we know that the vacuole is where we're having our water held. It's our central vacuole. I'm noticing that our pH in the vacuole is 6.6, and then once the water goes in, it's at 7.7. The color change goes from red to blue, and you can see the vacuole has a small volume to a large volume. So it tells us that there is this petal color of the Mexican Morning Glory changes from red to blue, and the petals swell during the flower opening. Pigment blue is found in the vacuole of the petal cells. Petal cell is determined by the pH of the vacuole. A model of the Morning Glory before and after flower opening is shown in Table 1. Okay, and I think my arm kind of rotated off there a little bit. I'm sorry. And I will try to figure out how I can post this online for you all later, but everything is available in that review guide. So the first part we have to do is identify the cellular component in the model that's responsible for the increase in pH of the vacuole. So noticing that our bud has a 6.6 pH and that our open flower has 7.7. And so what we see here is that there was an increase in the pH. So you have to think to yourself, okay, well, pH looks at the concentration of hydronium ions, or H+. Okay, and so in the picture, we can see that there's all these H+ ions. And so we have to think to ourselves, okay, well, what is causing my H+ to decrease? When I go from 6.6 to 7.7, I'm increasing my pH, which means that my hydronium concentration is decreasing. I'm getting less hydrogen ions. So what's causing my hydrogen ions to leave? Okay, so if you look here, you look at the picture, you see that, okay, well, in the vacuole, I have this pump that's bringing protons in, and I don't see anything happening here. But when it's open, I see that my hydrogens are leaving through this thing right here. It says that it's a K plus H plus transporter. So you can see right here that those ions are flowing out, okay? And so our first answer is that we're looking at the, oh, I'm sorry. I even made a little circle to help us with that. We can see that the K plus H plus transport protein. So you would have to mention in your response that the transport protein is what's allowing those protons to leave. And as those protons leave, they increase the pH. Okay. Now, in the question, it told you it was an identify question. Now, identify questions, and your teachers probably tell you the same thing. Identify means that you just have to give the one word. okay? But how hard is it really to make it a full sentence and ensure you get that point? So I would say the cellular component that increased the pH is the transport protein. And the K plus H plus transport, that little part that was in parentheses, is optional. You could have said it, you didn't have to say it. I probably would have written the whole thing just because the whole thing was given in the diagram. If they label something in a diagram, you want to give them that label back. You want to tell them that word because they want to make sure you understand exactly what it is. And what if there's two transport proteins on the diagram? So you want to be as clear about what you're trying to tell the readers as possible. So the second part of the question says that we're describing the component's role in changing the pH. Well, what did we see happening? Well, we saw those protons were leaving that vacuole. So you would have to say it allows the H plus ions to leave the vacuole, okay? Because we can see clearly in our picture, H plus moved out, okay? So that's the thing about this question. All you had to do was look at the diagram. If you could see in the diagram and you knew from yourself, okay, well, pH has to do with that the hydrogen ions decrease. All you had to do was look at the picture and see how did the hydrogen ions decrease? What allowed those hydrogen ions to decrease? Okay, and all you would do is look at the picture and you say, oh, hydrogen left, that increased my pH. It happened through the transport protein. Boom, you got 2 points just from looking at a diagram and giving them information from that diagram that you were able to interpret. So that's a part of this free response. And AP is all a numbers game. Get as many points as you possibly can get. Hi, Miss Kaufman. Get as many points as you possibly can get in this whole thing, okay? It's all about points, okay? So part B says that the researcher claims that the activation of the K plus H plus transport protein causes the vacuole to swell with water. Provide reasoning to support the claim. So before the transport protein, we have small. After, we see that it's large. Notice there's no K plus in here, but that K plus H plus transport protein brought K's in. It brought some ion into our vacuole, okay? And whenever you have an increase in solute, it's going to cause the water to move in, right? Because it makes the environment hypertonic. And so if it's hypertonic, the water is going to flow from where it's hypotonic to where it's hypertonic. And so we're going to see that the water flows into the vacuole, which is why your vacuole got larger, was because of the fact that water flowed from hypo to hyper. Okay, so we said to provide reasoning. Oh, I didn't flip the camera. Sorry, guys. And so you should have said that the concentration of the solute increased, which you could clearly see there was no K plus here. Now there's K plus there. So the increase of the K plus or the solute. And you could have all said that the solute is moving into the vacuole, making it hypertonic. Or you could have used saying the lowers the water potential. You could have said any number of those things. And you just had to give one of these two things. There's 2 bullets here, but only one of them was required. So I hope that the first response was helpful. Okay, remember, if you have any questions, you can pop them into that little question box. And I'm more than welcome to help you and kind of answer them as we go. Our next question that we have is the 2018 exam number 6. And this one's looking at after you've produced a different protein, where it's going to end up in the whole cell. Okay, and so for 2018 number 6, we have cystic fibrosis, genetic condition that is associated with the defects in the CFTR protein. The CFTR protein is a gated ion channel that requires ATP binding in order to allow chloride ions to diffuse across the membrane. And so what you have to do is draw an arrow to provide the pathway for a production of a normal CFTR protein from gene expression to the final cellular location. Wait a minute. I have to draw an arrow that shows the path from where it's expressed to where it finally ends up. Okay, wait. So let's think for a second. Where in the cell do you express your genes? So we know that the DNA, the nucleus, okay, is where the DNA is. That DNA is transcribed and makes RNA, right? Have we started that expression part? Not really. We haven't expressed what's in the genes yet. So where does the RNA go? So the RNA is going to go to the ribosome. So the first part where we're seeing this whole thing process, okay, is that we see that we're going from the nucleus to the ribosome, And then after that, we see that it's going to go to the Golgi to be modified and packaged. And then finally goes to the plasma membrane, because we know that it's going to end up being in that plasma membrane because it allows things to diffuse across the membrane. So you should have drawn a picture showing an arrow going from your nucleus, which is where we made the mRNA, right? Going to the ribosomes, or you could have read the rough ER, didn't matter to them. Then going to the Golgi, and then going to the plasma membrane. Now you have to be very careful that this arrow does not extend past the plasma membrane. If you extend this arrow past the plasma membrane, you're showing that the material has been released from the cell. But in actuality, it went and is housed in that plasma membrane. So it's very important how you draw your pictures. So you could have just drawn a straight line going through, or you could have just done each one separately. So the nucleus, where we have our transcription translation, going to our ribosome. After our ribosome, we then go to our Golgi, then we go to a plasma membrane. Okay, so I hope that was logical. And you could have went to a ribosome that wasn't on the ER, but you had to come back to the ER in some way, because the ER is how we get it to that membrane. So the second part B, is about identify the most likely cellular location of the ribosomes that synthesize the protein. Okay, so since we know that it goes to the membrane, which of these spots do we see this labeled? Okay, where is the ribosome that allows it to get to the membrane? Well, logically, that would be your endoplasmic reticulum. You could have said rough ER, but all you had to say was actually just say the endoplasmic reticulum, or you could have just said the ER. Okay. Now again, this was an identifying point. You actually on the exam would have gotten credit just by drawing the picture, and then just first line you wrote ER, and then the next line you could have read the part C. Okay. You could have gotten credit. But how hard is it to write a full sentence and ensure you get that point? I've seen identification points where you don't get it unless it's in a complete sentence. There was a question, it was a genetics question, that you had to write out the ratio as a sentence. The ratio is. So in this case, I would have written out the protein is synthesized in the rough ER, or the protein is synthesized in the ER, the protein is synthesized in the endoplasmic reticulum. I would have made it a full complete sentence just to ensure I got that point. Part C, in this one, we want to identify the most likely cellular location of the mutant protein that has an amino acid substitution at the ATP binding site. Well, it's still going to act as an ion channel, right? It still is going to be an ion channel, and it's going to allow things to diffuse across the membrane. Well, if it's diffusing something across the membrane, logically, it's in the membrane. The final location of it is in the membrane, allowing things to move across. So, again, this was another identification. So you could have just written out plasma membrane or just written out membrane, and you would have gotten credit for this, okay? But how hard was it to write a full sentence saying the protein will result in the plasma membrane? The protein will be in the plasma membrane at the end. Like, make it a full sentence to ensure you get that point. So again, if there's any questions, you should pop them down to the little question box, just in case you have any, just remember that. So our next question that we have is the 2017 question. It's number 8. So it says that estrogen is a small hydrophobic lipid hormone that promotes cell division and the development of reproductive structures in mammals. Estrogen passively diffuses across the plasma membrane and binds the receptor proteins in the cytoplasm of target cells. Describe one characteristic of the plasma membrane that allows estrogen to passively cross the membrane. So in the prompt, they tell me that the estrogen is hydrophobic. Well, how does a hydrophobic material pass through an area? Well, it has to pass through something that's also hydrophobic. So we all know that the plasma membrane is made up of phospholipids. And we know phospholipids are going to have a polar and a nonpolar region. The polar region is that phosphate head, and the nonpolar region would be the fatty acid tails that sit in the interior of the membrane. So we have phosphates facing the extracellular and intracellular regions, and then the interior of the actual membrane, like the interior of the membrane, is going to be nonpolar due to those hydrophobic tails. And so you would talk about that our plasma membrane is hydrophobic because, of course, we allow this hydrophobic material to pass through it. Or you could have just said that it passed through in between the spaces of the phospholipids. That was another potential answer you could have put here. So all you would do is make the connection to why can this estrogen, as hydrophobic, why and how is it able to pass through the plasma membrane? Well, plasma membrane must also be hydrophobic or nonpolar to allow that process to take place. Okay. So again, if you have questions, pop them in there. So part B is supposedly no longer in the standards. When I looked it up, I believe it didn't actually show to be in the standards anymore, which is weird to me, but that's okay. So it says in a laboratory experiment, a researcher generates antibodies that bind to purified estrogen receptors extracted from cells. The researcher uses the antibodies in an attempt to treat estrogen-dependent cancers, but finds that the treatment is ineffective. Explain the ineffectiveness of the antibodies for treating estrogen-dependent cancers. Wait. Let's think about it for a second. So we know that, that's not what I meant to do. Where'd I go? I pressed too many buttons. Okay. So the antibodies, I thought I made a little underline, but I guess I didn't. Hi, Marco. The antibodies are produced by a cell, and they're actually proteins. And so we know that our proteins are going to be polar. And so logically, if we know that the receptor is inside of the membrane, the only way that the estrogen antibodies, these antibodies are supposed to bind to that receptor, the only way they can get through is if the antibodies are nonpolar. But the antibodies must be polar, and that's why they're not able to get through. And so the reason why the antibodies are ineffective is because the antibody can't get through the membrane because it's polar and the membrane is nonpolar. It cannot reach the receptor to bind to the receptor to act as an inhibitor. And so you could have said that antibodies are unable, I didn't flip the screen, sorry. The antibodies are unable to cross the cell, and you could say that they're unable to cross because of the fact that we know that they're polar. Or you could say that the antibodies are not going to bind to the receptors, okay? So you didn't actually have to know about the actual polarity of it, but you did have to make a connection that the reason why these antibodies are not effective is because of the fact that they can't get into the cell to get to those receptors. And since they can't get to the receptors, they can't bind to them to inhibit the estrogen from binding to those receptors. So again, if you have questions, pop them down in the box. I believe we have one more question we're going to go through. And this one was actually from 2013, so it's kind of as far back as you probably could go to get a similar question to how we're going to see it come May or June, depending on when you're taking your exam. So somebody just said you need both of those bullets for that full 2 points. Yeah, you need both of those points. This one was number 8, and so on the old design that was a 3-pointer, and so part A was 1 point, and so part B was 2. Yeah, you had to have both of those bullets in order to get that credit. So you had to tell me that they were unable to enter, and you had to tell me that they were unable to bind. Thank you for making that clarification, Lauren. So fourth question. 2013, number 6. So in this question, you're welcome. This question, we are given information about 3 different cells. And so we see that we have cell X, Y, and Z. And so they tell you different organelles. They tell you the smoothie yard, they tell you the roughy yard, the mitochondria, the cilia, and the Golgi. And you have to identify the function of the cell and explain how that data supports the identification. Okay. And this is a number 6. And so that means that there's going to be 3 points here. So you could make the connection and say, okay, well, I have to have function and explanation together to get my point. That kind of helps you to see, okay, this has to be one grouping together. So first thing, let's think about our different organelles. So smoothie R's main job is to detoxify the cell. It also has a job in which it's going to store calcium. It also creates the membrane for the cell. Roughy R is going to be responsible for making our membrane proteins or our proteins for secretion. The mitochondria, don't you dare say powerhouse of the cell in your brain right now. You know that that is not what teachers want you to say. Is going to be a site of ATP synthesis. So in the mitochondria is where we're going to have ATP synthesized. Cilia is going to be a, you can hear you're going to hear my son in the background. Sorry, guys. Sorry, penguins. The cilia is going to allow for locomotion. It allows the cell to move or allows things to move past our cell. And then last but not least, our Golgi is going to package and modify the different products from our rough ER. And so it's not illogical that since there's a large amount of rough ER, there's also a large amount of Golgi. So thinking about this, let's look at just X. Cell X has a large amount of mitochondria, and it also has cilia present. So this means that we must have some type of cell that requires a lot of energy and allows movement. So whether we're looking at a cell that itself moves, or if we're looking at, say, like, for example, your trachea, right? Your trachea has mucus all in it. And so the cilia moves that mucus up so that it goes down into your esophagus, because this mucus is going to trap different pathogens and different things that you breathe in that you don't really want to get down into your lungs. And so an example of this could just be a cell that's going to transport, or a cell that's going to kind of move something. And so the answers that they had on the prompt, you could have said that it was responsible for locomotion or movement or surface transport. And then you had to mention that we had cilia for movement, and you had to mention the large amounts of mitochondria that providing the energy for the locomotion or the movement past the cell. Okay, I will tell you, this is a loaded question. You had to give a lot of detail to get that point. So be very, very careful and count your points. You know that your first 2 questions are going to be 8 to 10 points, and you know that your last 4 questions are all going to be 4 points. So you can kind of count your points and make sure that you're giving enough information to get your points. So going into our next quote part. So if we look at Y has a large amount of smooth ER. It has a moderate amount of mitochondria and a large amount of Golgi and a large amount of rough ER. Okay, so you could probably go 2 different ways on this. You could look at the fact that it has a large amount of rough ER and a large amount of Golgi and say, oh, well, this cell must be for secretion. It must be in order for, say, like we're thinking about like your pancreas that makes your insulin, right? So this is in charge of producing some type of protein that needs to be secreted from the cell. As you see here, we have our rough ER to synthesize and our Golgi to package. You could also take the path of maybe it's for detoxification. Due to the large amount of smooth ER, this cell is going to detoxify like your liver does. Your liver's job is detoxification. So, of course, you have a large number of smooth ER in your liver. So let's see what the options that were on the rubric or scoring guidelines. You could have said secretion or protein synthesis, and again bringing in the large amount of rough ER and Golgi to produce a package. Or you could have said that we had detoxification or lipid synthesis because of the large amounts of the smooth ER. So you could have taken either of those approaches to get it. And again, you have to get this and this, or this and this. If you went diagonal, you would not get credit because of the fact that the detoxification has nothing to do with the rough ER and the Golgi. Same thing as the secretion has nothing to do with the smooth ER. So you had to demonstrate to the readers that you understood the function of the organelle and how that gives function to the cell. And now Z, which I find to be a trick question, but it is what it is. Z has no smooth ER, has no rough ER, has no mitochondria, has no cilia, and has no Golgi. Ooh, what did I just do? I don't know, I pressed the button and something turned colors. Anyways, it has none of the organelles. Ooh. What cell has absolutely no organelles? That's mind-blowing. There are cells that don't have organelles? Yes, there are cells that don't have organelles. An example of which would be your red blood cells. Red blood cells don't have any organelles. Why? Because we want more surface area. The more surface area you have, the more hemoglobin, and thus the more oxygen that red blood cell can carry. Another example would be xylem, the cells that make up the xylem, the tracheids in your plant cells. These are going to allow us to move the water, okay? Or a dead cell. If the cell's dead, then it has no organelles. So let's look at the different options that were available on the prompt, the scoring guidelines. You could have said that it was for transport. And of course, it doesn't require the organelles. You can talk about protection, and again, does not require the organelles. Support, and does not require the organelles. Storage, does not require the organelles, or it has no function because the cell is dead or going through apoptosis. So, as I said, funky question, and it throws you through a loop. You sat there probably thinking to yourself, what cell has no organelles? And so you just had to make sure that you understood that maybe it didn't have function because of the fact that maybe the cell's dead and it has no function because it doesn't require any organelles. So any last minute questions before I log off? So quick reminder, we will do this again next Friday. 2 o'clock seemed to work okay. My son is still sleeping, so I guess we're good there. Actually, who's talking a little bit? Maybe my mom's still here watching him. So next Friday, we will be meeting and we're going to go over these 2 FRQs. I believe they're Unit 3 FRQs. So I hope that today was helpful. Please give me any type of feedback so that our FRQ Fridays can be more helpful if you're not finding them to be helpful. And I hope you have a wonderful day. Bye, penguins.